Okay, class, hi, welcome to week 2. We are going to be going over chapters 2 and 3. This covers your basic chemistry and your macromolecules. This part of the class is designed to work with people who have a little bit of background in chemistry, but also for those who have not been exposed to chemistry at all. So we're going to touch on the surface of a lot of the subject matter and not dive too deep, but set a good baseline. And so just to set the precedence here, you might wonder, why do I need to know this? Why does this even matter? And I'll give you just a little bit of information. So in the book, you may or may not read the overviews at the beginning of the chapters. They have some cool facts. I personally always like these because it gives you like a real world application for some of the concepts that you're going to go over in each chapter. In this particular case, for the chemical basis of life, we go over the subject of ocean acidification. And if you take a look at our planet from space, you notice that we're on a water planet, right? You've heard this before. In fact, we're made up of about 71% water. And we typically take water for granted. The impact of climate change has started to begin gaining awareness. The primary cause of climate change is the release of carbon dioxide, or CO2, into the atmosphere by burning fossil fuels by humans. Not only does that CO2 contribute to global warming, but it also can dissolve really easily in water. Whenever it dissolves in water, it forms something called carbonic acid. So the way that the bonds and things work, which we're going to go over, creates this carbonic acid. Carbonic acid gets into the water and it causes things that make shells to not be able to make a shell that's strong enough. And this has a sort of what I call a ripple effect. So it helps form the shells like in crabs, lobsters, and clams. And as the ocean gets more acidic, those organisms have a harder time forming the shells and then other hard structures so that it can really easily breakable, which can be dangerous. That could harm the animal. There's a careful balance in the ecosystem, and whenever things get thrown off, again, there's a ripple effect. So it is important. In this chapter, we're going to look at the importance of water as life as we know it, and then also the atoms that are responsible for making up those compounds of life. So we're kind of getting down to the basic level of what life is. So we're going to start with the basics. We start with matter. What is matter? So atom, that's the smallest unit of matter. They cannot be further subdivided without losing its essential properties. So that means that atoms are individual components of an element, and then atoms with different properties have different elements. An element is a substance that cannot be broken down chemically into any other substance. So for example, water, H2O. You have 2 hydrogen and 1 oxygen. Those are the elements that make up water. The hydrogen and the oxygen, those can't be broken down any further. So what makes an atom? So we know that there is a nucleus, that is the center of an atom. So we're going to kind of break down what they look like, how they're made. I'll go ahead and populate all of these here. So you have a neutron. This is your neutral particle. This has no electric charge, nothing's happening, hence the term neutron. And then we also have protons. Protons are positively charged particles, and the number of protons in the nucleus determine the element's atomic number. So if you have an atomic number of 5, you have 5 protons. Protons and neutrons have similar mass. They make up some of the atomic mass of an atom. So actually combined, the atomic mass is the combination of protons and neutrons. The cloud or shell around the nucleus is where our electrons live. So those are negatively charged particles. They were very, very small. They don't really have a weight. They don't contribute. Almost nothing do they contribute to the atomic mass. If you take a look at the picture here, this is a image underneath an electron microscope. It's a very, very high magnification. And you can see this is gold foil, and you can actually see the individual atoms. It's so magnified. So the basic structure of your atom, one thing, there's a balance that's kept here. So electrons are always in motion, and if you have one proton of a positive charge, then you have one electron of a negative charge. So that being said, you have your hydrogen atom and then a carbon atom as an example here. So for hydrogen, you have one proton, no neutrons, and one electron. But for the carbon atom, you have 6 protons, 6 neutrons, and 6 electrons. So the forces of attraction between the positive and negative charges, that's what keeps the electrons close to the nucleus. So here we have our periodic table of elements. We kind of looked at carbon on the previous stage, or the previous slide, so we're looking at carbon here as well. Some of the periodic table's elements are synthetically made, some of them are natural. We're not going to concentrate on too many of them, but right here we're going to be looking at carbon. Closer look at carbon here. So you can see we have carbon 12, carbon 13, and carbon 14. Why do we have 3 of these? Well, we have atoms with the same atomic number, same number of protons, but different atomic masses. We call those isotopes. So carbon 12 is a form of carbon that's found naturally occurring in the environment. We get the atomic mass down here at the bottom of 12 from the protons and the neutrons. So we have 6 and 6, 12. For carbon 13, it actually has 7 neutrons, so you get an atomic mass of 13. For carbon 14, it has 8. It makes it a little bit unstable. So it has an atomic mass of 14. Now, carbon 14 is actually the form of carbon that they use for carbon dating because it is radioactive, but it has a stable decay. So they can measure based on the amount of decay. They can age fossils, certain things like that. But these are isotopes, and their atomic mass can be a little bit different. Everything is made up of a combination of about 90 or so naturally occurring elements. For us, the big 4 of our bodies, about 96% of our body are composed of just 4 elements. So the highest amount, no surprise, we have oxygen at about 65%. We have carbon at about 18.5, and hydrogen at 9.5, and nitrogen at about 3. The other remaining 4%, calcium, potassium, sodium, phosphorus, sulfur, and chlorine, trace amounts of these, that's less than 0.1% of 15 other elements are also found in the body. So we've got our big 4, a smaller 6 divided up between the 6 of them in that 4% range, and then very, very low amounts of other things. So the big 4 is what really matters here. That's the takeaway from this slide. We have a couple of different types of chemical bonds. We have covalent, we have ionic, and we have hydrogen. And the difference between those things lie in the electrons. It's all about the electrons. I always find it helpful to have a visual when it comes to looking at electrons and protons and neutrons and things like that. So for the different types of chemical bonds that involve electrons, we're just going to look here. So we have chemistry, that's just the study of electrons. The electrons are what dictate the bonds. So for example, oxygen is considered unstable and it wants to bond. And the reason that is the case is because if you look at the outer shell, it only has 6 spots filled out of its 8. So there's some vacancies. The first electron shell's capacity is filled. It has 2. It's good to go there. But when you break it down to the outside, there's 2 spots missing. So it's looking for something to bond with. Now, as you might have figured out, atoms are more stable when their shells are filled. So their outermost electron shell, the goal is to have it full. So stable atoms tend to not react or bond with other atoms. So your unstable hydrogen is unstable. It's got something missing in its outer shell. And the stable example is the helium atom, which has its shell filled. Now to get into the definitions of each of the different types of bonds. The first one is covalent. This is where molecules that share valence electrons actually will combine to stabilize their electron shells. So think of it as a seesaw, right? You've got one person on one side, you need someone on the other side to make it stable. That's kind of what hydrogen atoms do. So they each have something missing. They have one each in their outer shell. So they combine together and they use both of those together to technically each have 2. And that's where you get your H2 molecule. It likes to bond together, and it's kind of this stabilizing action, and we call that a covalent bond. You can also get something called a double bond. This is where 2 electrons are shared between 2 atoms. So like in the previous slide, we had 1 or 2 shared, but both atoms need 2 electrons to be stable, right, in this case, and so they combine together. So you end up with a double amount because there's 2 vacancies on each instead of just 1 vacancy on each. They will still combine together and essentially have the same effect, but we call that a double bond. A double bond is stronger than a single bond. So this is O2. This is a very stable molecule in this case. Now with an ionic bond, it's a little bit different. So take a look at these 2 molecules. You have sodium and chlorine. Are they stable? Why or why not? And in this case, who's going to donate an electron and who's going to be the acceptor? Sodium is going to donate the electron because if you look, it has one floating kind of out there, just an extra in its very outer shell. But the chloride ion is missing one. So it's very obvious. So sodium is going to donate its electron. So officially an ionic bond is defined as when one atom transfers 1 or more electrons completely to another atom. The result is these 2 oppositely charged ions. And we've all heard the term opposites attract. Well, in chemistry that actually is the case. So the 2 oppositely charged ions will attract each other and form a compound. And that's when a molecule is made of 2 or more elements, like this here, this sodium chloride. They've combined together, and you get a compound element. So NaCl, sodium chloride, this is salt. It's just salt. It's the salt that is most responsible for the salinity of seawater or extracellular fluid of multicellular organisms. And then the last type of bond that we are talking about is a hydrogen bond, which, you guessed it, involves hydrogen. So hydrogen is bonded to an atom as a covalent bond. You have 8 protons, 1 proton and 1 proton on the hydrogen, they're bonded. But the extra protons in the oxygen nucleus cause the shared electrons to spend more time near the more positively charged nucleus. This causes the oxygen to be slightly negative and the hydrogen to be slightly positive. So then the hydrogen bond, that's an attraction between the slightly positive charged hydrogen atom of the one molecule and the slightly negative charged atom of another, as we were talking about opposites attract. So it is an attraction that causes a bond, as in the water molecule. So you have H2O because of the hydrogen bonds in the water. So in summary, you have your three types of bonds. Covalent, a strong bond that's formed when the atoms share electrons in order to become more stable, forming a molecule. So that bond strength is strong, as in the H2 molecule. An ionic bond is an attraction between two oppositely charged ions that form a compound, as in the salt compound. That bond strength is also strong. For a hydrogen bond, that's an attraction between the slightly positive charged hydrogen atom of one molecule and the slightly negatively charged atom of another. That bond strength is weak. A hydrogen bond is typically easily broken. And on that note, we're going to transition into the abilities of water. Because of the hydrogen bonds, water has unique properties. So it has cohesion, has a large heat capacity, so it can hold on to heat for a long time. It has a low density as a solid, and it's a really good solvent. Cohesion is the surface tension of water. You may have done experiments where you set a needle on a slightly overflowed glass of water, and it doesn't overflow the cup if you're careful because it has the cohesion, the surface tension. So that is from a hydrogen bond, which is a V-shaped water molecule that are held together by the hydrogen bonds. And they're just strong enough to give the water a surface tension that acts similarly to a net. And this is how boats float. Or you see the water bugs, that kind of water gliders that go across the surface of the water. Whenever I'm swimming in my pool in the summer, the first time it happened, it really freaked me out. But red wasps or yellow jackets will come and they'll land on top of the water and they spread their feet really wide so they can get a drink. And as long as I don't splash or freak out, they won't sting me. They just get their drink and they fly away. But they land with their legs kind of like spread as far as they can go to take advantage of the surface tension. Cohesion also works as a siphon. So whenever we have evaporation or transpiration happen from plants, that's because the sticky connection of the hydrogen bonds, it's the water, because when the water is pulled up through the plant, the hydrogen bonds being sticky is what allows it to continuously flow upwards. And then the water molecule is released into the atmosphere at that point. So they're pulled into the root system, and then through the siphon, they go up through the stem of the plant and then out of the leaves. Evaporation also plays a role in heat capacity. So the heat or the energy from the sun disrupts some of the hydrogen bonds that are formed between the water molecules. New hydrogen bonds are formed almost as quickly as they are disrupted, though. So you can see the red dotted line is the disrupted bond and the blue is newly formed, and they're all over the place. But for example, while the sun's energy might make sand really hot, when the same energy hits water, most of that energy breaks the hydrogen bonds, which might later reform. But rather than increasing the water's temperature, it doesn't. So it can maintain because of the way the heat disrupts the hydrogen bonds. Another interesting property of water is that its ability to be less dense. So as a liquid form, as in the picture here, you have a can of soda. In liquid form, you got plenty of room in the can. But if you were to freeze it, then the water expands and it busts through the can, and then you have a mess that you have to clean up. So why does ice float? The density of water, this is what we're getting into. So liquid water, the water molecules move about freely. They can kind of ebb and flow, allowing them to be closer to another. Whenever you freeze that, the hydrogen bond arranges the water molecules into like a lattice, there's a pattern, a crystalline lattice that keeps them slightly farther apart. So they can't move in close and then move far away. They can't flow. It just freezes them in place. And so it always makes this pattern, and it keeps them farther apart. And so that's how it expands, and it becomes less dense as a result. Water also works really well as a solvent. So a solvent is the solution that something is being dissolved into. The solute is what goes into the solvent. So in this case, the solute is the table salt, NaCl. That's the crystal of salt. And then you place it into the water. So when it's placed in water, the ionic compounds like NaCl are broken down into, they're two separate ions. So you have the Na, your sodium, and then your chloride, which are both positive and negative. So the positively charged sodium ions are attracted to the negatively charged side of the H2O molecule, while the negatively charged chloride ions are attracted to the positively charged side. So they're drawn to the pieces of the water as they split apart. You can also measure how acidic or basic a solution is. We all know pH. So you have your non-ionized water molecule, H2O. You have an ionized hydrogen molecule, and then an ionized hydrogen molecule. So whenever you're going to measure pH, if you have excess hydrogen, that equals acidic. If you have excess OH, an oxygen-hydrogen bond, then you have a more basic solution. And if you have equal amounts of hydrogen and OH, then you have a neutral pH. So you have your alkalinity if you have too much OH, your acid if you have too much hydrogen, and then your neutral balance between the two. Acidic fluids that have a greater proportion of hydrogen ions to OH ions, those hydrogen ions are very reactive. Strong acids are corrosive to metals. Acids break down food in our stomachs, in our digestive tract, and they generally have a sour taste. On the other hand, bases, they have a greater proportion of the OH ions than hydrogen. They bind with hydrogen and neutralize acids. That's what the OH ions do. Strong bases are caustic to your skin. If you've ever gotten into a pool that is too alkaline, it can make your nose burn, your skin can react, and you can get a rash. It can be damaging, but they use it a lot in many household cleaners because it can really cut through some things. They are generally a bitter taste and slippery. The next subject matter is macromolecules. So we have large molecules that are made up from smaller building blocks or subunits. The macromolecules are essential to building and functioning of any living organism. You have four types of those. You have carbohydrates, lipids, proteins, and nucleic acids. So first up we have carbohydrates. I absolutely love carbs. Bread, pasta, you name it. The most terrible things are delicious and usually are carbs. But for carbohydrates, technically, the definition is they're primarily composed of carbon, hydrogen, and oxygen. They're fuel for cellular activity and form cellular structure. They have carbon and hydrogen bonds of carbs. They store large amounts of potential energy. They're easily broken down by organisms, so you can break the bonds and use that as energy for your body. You have several categories based on size and composition. So you have monosaccharides. Those are simple sugars. There are 3-6 carbon atoms. They're not usually carbs whenever they're broken down. You have a disaccharide, which is a monosaccharide plus a monosaccharide, and then so obviously you have two. There are 2 simple sugars that are bonded, and then a polysaccharide or a complex carbohydrate. There are 3 or 10,000 sugars bonded together, so you have a lot of combinations you could have here. So the bonds between simple sugars that are formed, glucose and fructose, you can see this is like a monosaccharide. The disaccharides are formed by the union of 2 simple sugars. So sucrose, a table sugar, is glucose and fructose. So those are 2 sugars bonded together. And then your complex carb, a polysaccharide, that's formed by the union of many simple sugars. Like starch, that consists of hundreds of glucose molecules, for example. Some of the common monosaccharides are glucose and fructose, which are oddly enough both C6H12O6 for their chemical formula, but they're just put together a little bit differently. So the difference is, so you have glucose, that's found in tree saps and in the roots of plants, but then fructose, that's found in fruits, vegetables, and honey, and it's the preferred taste by animals, even as humans. I love fruits. I love the sweetness of honey. I have no problem eating my vegetables. I can definitely say it's my preferred taste, although I don't make a habit of tasting tree sap or on roots of plants, personally. When you have glucose in your blood, it has three fates. So glucose goes into your bloodstream. One path, you have energy that's needed. Those molecular bonds are broken down. You get new lower energy bonds are formed, and you get fuel for energy. The other pathway is when energy is not needed, if you're sleeping. So you have energy storage that is happening. You have short-term and you have long-term. So you have short-term, it's stored as glycogen, and then long-term, it's converted to fat. So this is why you don't want to eat a lot of high sugar before bed. It's going to end up as fat. So glycogen as short-term energy, that's stored in your liver and in your muscle. So it's the first energy source that's tapped into whenever you are reducing your calories. Excuse me. So a pound of glycogen has about 4 pounds of water molecules bound to it. Dieters lose a lot of water weight, quote-unquote water weight, during the first days of a diet because of this structure. Take a look at the way digestion happens between disaccharides and complex carbs, polysaccharides. You get energy either way, but for polysaccharides, that's the union of two simple sugars, so glucose and fructose, so you get sucrose. The bonds between those simple sugars will get broken. They get broken down further into their two individual sugars, and then those break and give you energy, so you could get energy from both of them. And then for the complex carbs, the polysaccharides are many simple sugars, so like starch, hundreds of glucose molecules, and you break those down and you just get energy from the one. Both forms of carbs will give you energy, but the difference is the length of time it takes for them to break down. So whenever you eat something like fructose, your blood sugar spikes right away, and then over time it slowly dips. But for complex carbs, that kind of rises to a peak and then steadies out, and you get a more steady long-term processing. So complex carbs are typically desirable as the energy source for someone who may be dieting or working out. You don't really want the blood sugar spike. You kind of want more level energy. You also have non-digestible carbs. For example, cellulose, that gives trees their rigidity to grow tall, and it makes wood a desirable building material. Chitin offers protection for lobsters. Those are the things that you don't really want to eat. It's also in vegetables and the walls of vegetables, so this is like your fiber. These are things that your body won't digest. It'll pass through, and that's how it kind of like cleans everything out. Ahead of myself. But as I was saying, so cellulose in human diets is known as fiber. It's non-digestible. So what is the benefit? So it's indigestible, but it aids in digestion, and it has a lot of other health benefits. Now we'll move on to lipids. These are the fatty compounds. The typical features of lipids include their non-polar molecules that do not dissolve in water. They're greasy to the touch, and they can be used for energy storage. So the three types of lipids, you have fats, you have sterols, and you have phospholipids. So the function of fats, they're long-term energy storage and for insulation. Think of blubber for like a seal or a whale. Sterols, they regulate growth and development. And then phospholipids, those are what make up our cellular membranes. When you take a look at fats, they have two distinct regions. So you have a head, a molecule known as a glycerol, and then a tail. Those are known as fatty acids, or long. If they're long, they're called hydrocarbons. They're nonpolar. So you can see you have a head and you have the fatty acid tails. The symbol for fat, a triglyceride, this is what's used in the book. over here on the right. Fat in our food, when you eat it, it's known as a triglyceride. So you have 3 fatty acids that are solid at room temperature, called fats. You have liquid at room temperature, called oils. And so for oil as a lipid and water, they don't mix. So the lipids, they actually have non-polar tails. Non-polar means hydrophobic. We know phobias mean a fear of something, so they don't like water. They have a fear of water. But then you also have hydrophilic heads. Those are water loving. So a hydrophile is a water lover. What do you prefer? And why? Why do we love fat so much? It gives you more energy, bottom line. So 1 gram of fat is about 9 calories of energy, and 1 gram of carbs is about 4 calories of energy. I think it was during Shark Week, I can't remember exactly, but I believe it was 1 pound of whale blubber. If a shark ate that, that was about 4,000 calories of energy. So they didn't need to eat very much of it. Our ancestors, they experienced food shortages regularly throughout history. This is something that you'll find once you start reading. Fats are the highest source of calories, and then individuals that ate more fats would have a better opportunity to store and keep that energy and survive the famine. Also, it will cause a taste preference towards fat. So our bodies are set up to survive, which generally involves getting as many calories as possible. So we crave this. And it's not very beneficial to us in an environment where food shortages are rare and fats are readily available. Are good fats and bad fats. So for dietary fats, you have saturated versus unsaturated. Saturated being the not so healthy of the two. So that is where each carbon atom is bonded to 2 hydrogen atoms. That's the maximum amount. So you have animal fats, cheese and meat. Those are solid at room temperature, but they're not essential to human health. So they could lead to things like heart disease and strokes. And for unsaturated, those are some carbon atoms only bond to 1 hydrogen atom. So that's where we get our plant fats, avocados, peanuts, olive oil, fish oil, that sort of stuff. Those are the healthy fat. They are liquid at room temperature, and they can actually help lower your cholesterol. So you actually do have good versus bad here. We also have this thing called hydrogenation. Hydrogenation is the artificial addition of hydrogen atoms to an unsaturated fat. This improves food's taste or texture or shelf life. So unsaturated fatty acid is in its cis form, and then partially hydrogenated trans fatty acid, that's when it's been changed, that's its trans form. So hydrogenation converts some of the double bonds to single bonds, but it adds hydrogens in the less healthful trans position, which changes the orientation of the double bond. So it can increase the saturation of fats for better and for worse at the same time. Sterols do not function in energy storage. They regulate growth and development. So they're all based on a structure that has a 4-fused carbon ring. They all look the same, or not look the same, but they're based on that same structure. Cholesterol is an important component of cell membranes in animals. So dietary cholesterol can attach to thicken the vessel walls, and if it thickens it too much, that can cause some pretty serious health problems. Steroid hormones, those regulate sexual development, maturation, and sex cell production. So estrogen influences memory and mood, and testosterone stimulates muscle growth. So these are all in the sterol family. Synthetic versions of testosterone can cause things like roid rage. We've heard of this. It can also cause high cholesterol. It can cause cancer, abnormal growth of cells, and then almost all athletic organizations have banned their use because of the damaging side effects. Phospholipids, these are what I was talking about earlier with the cellular membrane structure. We have phosphate heads and 2 fatty acid tails. Over here on the right, this is the symbol for the phospholipid that we use in the book. And you can tell they have a hydrophilic head, they love water, and a hydrophobic tail that is a water hater. It tries to get away from it. But they're the most abundant lipid in the cell membranes. You have a phospholipid bilayer, so there's 2 layers of these to get into the cell. It's very, very, very important for cell membrane and function. So in water, like I was saying a moment ago, hydrophilic water-loving heads, you can tell they're all around the outside. They stay near the water and they love it. And then the tails on the inside go away from the water, and they're always like this. They hate it. So the phospholipids align so that their heads extend towards the water while their tails, excuse me, are directed away from it. Waxes are complex lipids with long fatty acid tails. It has a head of a glycerol molecule, a tail of one long fatty acid, and they're strongly hydrophobic. You know this if you've ever tried to put wax in water, or you wax your car, so it keeps things from sticking to it. They're tightly packed molecules that result in the substance can be firm, but also water repellent at the same time. They provide a protective and waterproof layer in many organisms, like waxy leaves, duck feathers, and even in bees for honey. Next, we have proteins. These are the chief building blocks of all life. And this information, it's stuff that you would have heard before over the years. So for proteins, you have your hair, your fingernails, feathers, horns, cartilage, and tendons. It all takes proteins to keep these functioning. But it also helps control your cell activity and constitutes in some hormones. So they're regulatory as well as structural. They're also protective. They help fight invading microorganisms. They help you coagulate your blood. And they transport, so they can carry molecules like oxygen around your body. So they serve a lot of different functions. They are also what allows your muscles to contract, allows your heart to pump, and sperm to swim. So to have a healthy body, you need to make sure that you have enough protein. But what makes a protein? Amino acids, short answer. Amino acids, they are built in pieces, and then they combine together to make proteins, and those go to different areas of your body to do serve different functions. So you have a side chain. That's the unique part of the 20 amino acids. They vary in size and shape and in charge. So you have alanine, glycine, and tryptophan over here in the box, and you can see that they look very different. And that's because they serve different functions. They attach differently, and it's needed. So these are the amino acids that have hydrophobic side chains. You have valine, leucine, isoleucine, phenylalanine, and methionine. The next row is the hydrophilic side chain, so the ones that love water. You have aspartic acid, asparagine, glutamic acid, glutamine, and serine, lysine, arginine, histine, threonine. Then you have ones that are intermediate, so that's glycine, alanine, cytosine, tyrosine, tryptophan, and proline. In our diet, protein is an energy source. Proteins store energy in their bonds. And then the protein that you eat is used as building blocks for protein that you produce. So you break down protein in your diet into amino acids. Those amino acids are then used in growth, repair, and replacement of tissues in the body. So you break down the protein to get to the amino acid pieces, and those get sent off the different areas of your body for whatever function you need them for. You cannot produce all of the amino acids that your body needs. So you have complete proteins that contains all of the essential amino acids. Our bodies don't produce those, so we have to get them from food. Our diet, like milk, eggs, fish, chicken, beef. And then you have your incomplete proteins that contain some of the essential amino acids, and that's what you get from your vegetables, your fruits, and your grains. Here's just a list of the essential amino acid content of common food. So you have some that are complete versus incomplete. So what is present at optimum level is darkened in like a green in the circle, and then what's not present at optimal levels is everything else. So the optimal levels, those are determined by the Institute of Medicine's Food and Nutrition Board. So this just gives you a full breakdown of what you need. So if you want to get all of these histidine, excuse me, isoleucine, leucine, all the whole list, you can get all of those from milk, tofu, chicken, and beef, like a steak, which I personally love steak, so I have no problem with that. Lentils and white rice, you're missing one, but then apples and almonds, you only get some of them. So that's why we have to have a well-balanced diet. We can't eat just meat all the time. But if you're looking to get these essential amino acids here, you can get those mostly from the meats. Protein structure, the primary structure, the sequence of amino acids is what they call a polypeptide chain, similar to the sequence of letters that spell out a word. So when you break it down, you have, they are usually in triplets, in threes. So you have your amino acids, and then they're all bonded with peptide bonds. The secondary structure, once those are formed, is you actually have a corkscrew-like twist or pleated folds that form by the hydrogen bonds between amino acids and the polypeptide chain. Then you have your tertiary structure, the three-dimensional shape that is formed by multiple twists and bends of that polypeptide chain. So first it puts together, it twists up like a little corkscrew, and then that twists even further. And then based on the side change, those interact differently. And then your quaternary structure, you have two or more proteins that bond together. So it's kind of their step up. Now, the sequence of amino acids, a polypeptide chain is more than two amino acids joined together. So obviously a chain is something that's combined. The secondary structure, the chain folds due to the hydrogen bonding between amino acids. The two most common patterns are the corkscrew shape and a zigzag fold. Denaturation is a disruption of protein folding. So whenever you heat something up, the hydrogen bonds that give a raw egg white its shape is disrupted. So then we see a visual change in the egg white as the proteins unfold and they lose their secondary and tertiary structure. And then you get a cooked egg. So for normal protein, it's kind of it's the tertiary structure. So you have the zigzags and folds, and those all twist up around each other. So that's what you see here on the left. In an extreme environment of heat or pH, then those are disrupted. So the protein shape and the function then essentially denatures. It falls apart, and it can no longer serve the function that it is supposed to. That's exactly what happens when it comes to hair. So if you have curly hair and you want to straighten it, you put your straightener on there, you're applying heat, it's disrupting the folds, and you're making it a different shape. And essentially, if you have curly hair, you can use a chemical relaxer for a longer lasting straightening effect. But then also on the flip side, if you have straight hair and you want to curl it, you're using a curling iron, you're applying heat, you're affecting the folds of your hair proteins. So you can really see the difference when it comes to something like hair as opposed to on a cellular level, which we don't, it's kind of an abstract thing. You know that it's happening, but you don't really see it. Last thing that we're going to cover is nucleic acids. I know that this has been a long lecture, but we combined 2 chapters in, so here we go. So for nucleic acids, these store the information for your protein production, but they're also used in the production of protein. So they are made up of individual units, which we call nucleotides. Those have 3 components: a phosphate group, a molecule of sugar, and a nitrogen-containing molecule. You have 2 types of these, which should sound familiar. You've got deoxyribonucleic acid, which is DNA, and then you have ribonucleic acid. So for the detailed structure of nucleotides, everything has a sugar phosphate backbone, a sugar molecule, phosphate group, and it's held together by that backbone. You have your nitrogenous or nitrogen-containing bases, which for DNA, you have adenine, thymine, guanine, and cytosine. For RNA, you have uracil instead of thymine. But you have that sugar phosphate backbone. So this is the last picture that populated is what it looks like. You have the backbone, your nitrogen base is hanging off of that phosphate group, and they're all hooked together in a line. So for DNA, That is made up of a double helix. Those are two sugar phosphate backbones, which are, they spiral around each other and they form this vertical structure, which you've seen, images of in your life. They're connected by the bases that stick out from the sugar molecules, because you can see the line in the middle. The bases stick out on either side and they pair up together. So you have base pairs. Adenine always pairs with thymine, and guanine always pairs with cytosine. So you have A, T, and G and C, and that is always how DNA will pair. The double helix structure is caused by those hydrogen bonds between the base pairs. Oh, I got ahead of my picture here. Okay, so yes, DNA. Adenine always pairs with thymine. A and T always together, and G and C always together. That will never change. Each cell in our body contains the DNA necessary to code for every single protein that we have to produce. And there can be 3 billion base pairs. Now, RNA, ribonucleic acid, is a little bit different. It's the middleman in the protein building process in the cell. As far as you go into like the central dogma of biology, it's DNA, RNA to protein. So RNA is the middle child. There's differences from DNA. So the sugar molecule has an extra oxygen. It only has one backbone, so you don't get the double helix. It's single. And then you have a uracil base instead of thymine, so the T is taken out. So A and U always pair up, and cytosine and guanine always pair up. So this is a good picture of a difference between the RNA. So there's 3 important structural differences, right? The sugar molecule has an extra oxygen, and then it only has one of those backbones, while DNA has 2. And then, like I just said, uracil replaces thymine. As a function, like I said, it's a middleman molecule. It takes instructions for the production of protein from DNA and then moves them to another part of the cell and actually directs the building of the protein. So DNA is like, hey, here's the instructions. RNA is like, let me take those instructions to where they need to go. And then protein is like the factory that actually builds the protein, or the part where they take it to.